right. Hello, grade 12s. Okay, we're going to be starting chapter 19 today, which is a continuation on our genetics unit. This time, this chapter is called Beyond Mendel. So we're going to look at other non-Mendelian situations. And so let's get started. So in order to get started, we have to talk a little bit about fruit flies, otherwise known as Drosophila. They are, as it says, the darling of genetics. In university, I got to do lots of fruit fly genetic labs, and you always knew it was that time of year because you'd be walking through the biosci building and there would be fruit flies just pretty much everywhere. It's pretty easy to tell the males from the females, especially under a microscope. And fruit flies are awesome for doing genetics experiments because they rapidly reproduce. They make at least 100 offspring per cross, and they're reproductively ready to go at the age of 10 to 15 years old. And they are, as I said, easily to distinguish the males from the females, which will become important as we talk about this chapter. So Thomas Morgan, it's a good name to know, was an experimenter who was working with Drosophila doing genetics crosses, and he crossed a white-eyed male, so eye color in fruit flies. Remember that red is what we call the wild type, it's the dominant type. White is a recessive situation. And he took a white-eyed male, crossed it with a red-eyed female, and they were pure breeding. All the F1 generation had red eyes. Conclusion, red eyes dominant to white eyes, no big deal. That's normally what we would expect. He then crossed two of those individuals from the F1 generation. He took a red-eyed male and mated it with a red-eyed female. The F2 generation were through 3/4 red, 1/4 white. Conclusion, that's our expected ratio for the heterozygous cross. Again, no big deal. But when he looked closer, he noticed that in the F2 generation, only the males had those white eyes. All the females had red eyes. That was a big deal because that was not expected. So what was the conclusion? The conclusion was that Morgan believed that the sex chromosomes must have been involved, since only males seem to be affected with the recessive trait. Now, fruit flies have 8 chromosomes, 4 homologous pairs, and one of those pairs we know are the sex chromosomes, and the other ones are called the autosomes. So the X chromosome is represented here, the much larger chromosome, and then there's our much smaller Y chromosome of the sex chromosomes. They are only homologous, slightly homologous, enough to pair, but obviously not very similar in size, shape, or gene arrangement. So we remember that males are XY and females are XX. They're lacking that Y chromosome. So Morgan concludes that the eye color is governed by a gene located only on the X chromosome. And he was right. So we call these genes sex-linked genes. And 99% of the time, they're going to be on the X chromosome because X chromosome is necessary for all humans, right? And fruit flies. They've all got the X chromosomes, so you can't have very many genes on the Y chromosome. It can't be that significant, otherwise females would be in trouble because they don't have it. So the rest of this slide is simply going to show you how the cross works out. Now I want you to notice something, if you can see it very closely. When we do sex-linked crosses, we have to show the X's and the Y's. So we give the 2 X's to the female and we use the alleles as superscripts. And so a red-eyed female is going to be X big R, X big R. And a white-eyed male is going to be X little r, being recessive, Y. Then we do our Punnett square just like we normally would, but we need to show our X's and Y's. So of course we're going to separate the genotypes there as if we were forming the gametes. So of course we could do a simpler Punnett square where it's a 2x1, but that's okay. So notice how it gets filled in. And this is going to prove that all females have red eyes and all males have red eyes. But the females are what we would call called the carriers because they carry that white allele. But the males do not. So then if we take that F1 generation, remember we have a heterozygous female and a red-eyed male, and we fill in that Punnett square, and we look at our results, we're going to see, all the females are red eyes. Theirs are 1/4 white, but they're all male. And then half of the males have red eyes. Now, when we do the phenotypes for sex-linked, we have to include male and female in our phenotypic description. All right, so we can't just say 3/4 red, 1/4 white. We'd have to say 50% red-eyed females and 25% red-eyed males, 25% white-eyed males. So a sex-linked trait is defined as a trait governed by a gene located on the sex chromosome. The Y chromosome for now is simply for gender determination, and that gets activated for the embryo, helps produce testosterone, creates maleness. Almost all the sex-linked traits are on the X chromosome. We won't discount the Y chromosome because sometimes you can't discount that, because what if there's a test question and you're supposed to identify it as Y-linked? Now, there are over more than 200 known sex-linked traits in humans, 3 that we focus on. Color blindness, red-green color blindness, hemophilia, a blood disorder, blood clotting disorder. We have the Romanovs here, so a famous family influenced by hemophilia. And then Duchenne muscular dystrophy is another sex-linked trait. Now, a few questions here. Can we use terms homozygous, heterozygous? Well, yes, we can. However, for males, heterozygous doesn't make any sense because you'd have to be carrying 2 different alleles. But if it's X-linked, or Y-linked for that matter, males only have 1 copy. So we could use the term hemizygous, but we certainly can't say heterozygous, or homozygous for a male for that matter. Females, yes. Can a son inherit an X-linked disorder from his father? Well, no, because what do sons get from their fathers in terms of the sex chromosomes? A Y. So there's no chance that a male can give his son a sex-linked disorder. Can women be carriers? Absolutely, right? Because they have 2 X's, so they can carry, say, a mutated allele. However, can men be carriers? No, absolutely not. Men have it or they don't with their 1 X. Are sex lengths more traits in males or females? Yes, it does affect the percentage of gender differently. Now, usually when I ask my class, if you were here, and I ask this question, most students are going to say, well, females are more affected because they have 2 X's. So therefore, they've got a better chance of getting an X-linked disorder. Well, ironically, it's the males, because males, see, women with their 2 X's, they have a better chance of having a good dominant allele, right? And then if they have the recessive allele on the other X, it doesn't matter that much because the dominant allele shows up. For males, they're stuck with what they get. So if they get an affected X, that's it. So actually males are much more affected, about 10 to 1, than females are. Now we're going to carry on and talk about Barr bodies. So in a female, they have their 2 X's. So I'm going to paraphrase very quickly here. You can take the time to read these slides slower in the PowerPoint. That's up to you. But there is a scientist by the name of Barr, and he found out that in women's cells that have 2 X's, only 1 X is active and the other X is dormant. So that dormant X, which can be seen on a microscope slide, is called a Barr body, after Murray Barr, I believe, the scientist who determined that. So that leads to some interesting conditions, because it is possible then for that mutated X to actually be active and have the dominant X inactive. And so that results in certain situations like anhidrotic ectodermal dysplasia is a situation where sweat glands end up being produced or overproduced in certain parts of the body and underproduced in other parts. So kind of patchy. You kind of get this patchy situation, all right? So there's that situation. There's also the situation in calico cats. So in calico cats, or in cats in general, there's a gene for red pigmentation, and it's on the X chromosome, and it has 2 alleles. Now remember, there's other genes that give color, so it's a polygenic situation. But if we focus on this one sex-linked gene, it may be carrying a dominant R for red. If there's the recessive R, then you wouldn't see that red pigmentation. So male cats, if we looked at these genotypes, can be orange/red. It's not really bright red, it's more of an orangey color. Or black, right? If they have that recessive and you don't see that red color. Female cats can be orange or black as well. But when females are heterozygous, you might expect the dominant orange color, but that's not what happens. Because of the Barr body syndrome, you end up with the tortoiseshell, or what we call the calico color, because again, we're going to get this patchy situation where we see orange, we can see black, all right, because of the inactive. Some of the cells are inactive, and so we're going to see that orange, or we're going to see that black in a patchy way. Now, the last thing, guys, we're going to look at very quickly is, again, when we see a pedigree, what are some tricks and tips to determining what the situation is, what we call the mode of inheritance. There's really 4 modes of inheritance now that we've started to talk about sex linkage. So there's the regular autosomal recessive, like albinism. There's autosomal dominant, like Huntington's disease. Remember, so we've seen those pedigrees in the last chapter. But now we could see sex-linked pedigrees, where we could have a sex-linked recessive, which is the more common situation on the X chromosome. We would have a recessive allele that could cause a disorder of some kind. Or we could very rarely have what's the sex-linked dominant situation. So let's take a look at some example Punnett squares and review the tips and tricks. So this is an example of color blindness, which we know is sex-linked. So there's many, many more men that have red-green color blindness than women. So let's see how this would work. So here's a colorblind male with a normal female. Now notice that in their generation of children here, in the F1 generation, we don't have anybody affected but the shaded circles are females that now carry that allele. Remember, because that male would look like this, right? So would little c, y, and then our female is, because she's not shaded in half, must have 2 dominant alleles to be able to see color. So notice how every female receives the X from the father. Every male receives the Y from the father. So notice how we have no affected sons. But every female is a carrier because they can't help but get the X from dad. And then this female can only give a dominant allele. And so there you go. Now if we follow it along here, we'll notice that we do see many, many more males affected than females. There's only 1 female affected. Now, we would know that an affected female must have a carrier for a mother, or a mother that has the disorder, and a father that has the disorder. Okay, so that this father gives that recessive allele to the daughter, and the daughter had to get another recessive allele from the mother. So therefore, the mother has to be a carrier. So there we go. So that's an example. This is the same one. Okay, and then we've got that explanation, which you can take the time to read about if it's not very, very clear about why this would be X-linked recessive mode of inheritance. And so here we have 4 tips, tricks. Okay, so we know the mutant allele. If it's a sex-linked, it's carried on the X, recessive most of the time. So men who inherit the recessive trait are always affected. They're hemizygous. because they only have that one X. The other chromosome is a Y. There is no male-to-male transmission. So males actually receive that disorder from an affected or a carrier female. Because what are males getting from their dads? Ys. And then there are usually, almost always, more affected males than females, as long as versus a typical autosomal pedigree. Okay, so now why is there a little green star? Because that usually can't be the only thing that we will see. Because just by chance, you could have an autosomal recessive that affects more males than females, right? So it's not a guarantee, but it certainly helps. Okay, so just more words about autosomal recessive. Whoops, I'm going backwards here. Okay, all right, so now if we look at this, we can pretty much assume that this is going to be probably not sex-linked, because we see two females, three, so we don't see lots more males affected than females, and we can know it's recessive because remember, if you have unaffected parents and one affected offspring, that has to be recessive. Okay, that has to be recessive. And notice that two recessive individuals, all they have is the recessive allele, and so every child is going to be affected, but they're only getting recessive alleles. So again, lots of words to explain all that you can take the time to look through yourself if what I said didn't make sense. Another similar autosomal, this is also, I believe, autosomal recessive, right? Because again, we see two unaffected parents having affected offspring. Okay, let's skip this generation. It's not sex-linked because equal males, female situation. And remember that this female would have to have an affected father. So definitely not sex-linked. Okay, so again, we've got 4 characteristics of typical autosomal recessive. We've talked about this before in the last chapter. A parent does not have to be affected to produce affected offspring. 2 affected parents always produce offspring that are all affected. Males and females are equal, and we can skip generations. Okay, not a guarantee, but most likely autosomal recessive. Oh, and unaffected parents or affected offspring may be blood related. Now, autosomal dominant, okay, in order to express the trait, an individual only needs one dominant allele. So let's take a look. Here's an example. Notice the trait is not skipping generations, right? Like if it was there, then it would be skipping this generation, but it's not skipping any generations. We have, 2 males. We actually have more females than males affected, so it's likely not sex-linked. And every affected individual has an affected parent. So that's all leading to autosomal dominant, and of course, there's explanation for this for you to go through at your own pace. Here's another example, Huntington's disease, autosomal dominant. Notice we don't skip generations, right? Every affected individual has an affected parent, and again, females, males affected, so not likely sex-linked. Here's the reminders or the points to look at. At least one parent is affected to produce affected offspring. 2 affected parents can produce an unaffected offspring. Males and females fairly equally affected. Each generation is usually affected. And if a generation is left unaffected, then the transmission of the trait is usually stopped in that family line. We don't see it further down. Okay, now there's just, to end this, guys, there's just some very quick examples here. So here we don't think it's sex-linked, right? Because we've got like quite a few females and males affected. Do we skip a generation? Yes, we do. Since we see a skip in generation here, this cannot be dominant. So on those two facts alone, we most likely think that this is an autosomal recessive situation. And again, two affected parents, all affected offspring. So this is autosomal recessive. This one here. So we apply the same kind of idea. Here we see an affected offspring with unaffected parents. So it's got to be recessive. Is it likely sex-linked? Well, what's our numbers of males and females? So quite a few females affected, especially over here. So again, probably autosomal recessive. This last one, what do we see? We see now, this one's interesting because I want to show you guys that every affected individual has an affected parent, right? We know it's not recessive because, or not autosomal recessive because again, these parents, if it was autosomal recessive, every child would have the disorder, okay? So it's definitely not autosomal recessive. Now we might think, hey, I don't think this is sex-linked. I see 2 females, 4 males. However, guys, I've looked at this one carefully, and it could be sex-linked. It could be. But if it's sex-linked, it has to be dominant. It can't be sex-linked recessive. The reason it can't be, let's see, can we explain why this can't be sex-linked recessive? This male, if it was sex-linked recessive, would get an X from mom and would definitely, so it can't be sex-linked recessive because this male would have to be affected. But can it be sex-linked dominant? Yes, it can. So very quickly, so we could have an X big C and an X little c. We could have, and then this is an affected individual, so, but it's dominant, right? We're seeing it's dominant. Okay, so if it's dominant, this dad gives all the females, so all females have to be affected. We see an affected female, but the males maybe not, right? Because the male can get this X with the little c, males always get the Y from dad. So that's why we could have potentially affected or unaffected males. Then over here, same deal, right? And yes, and if the female's unaffected, we'd have to have 2 little c's. Males affected, big C, Y. Okay, so let's see here. Yeah, no, you know what? I think we just proved, because this male would just get a little c, and Y comes from dad, so this should be a normal male. So actually, I think I've proven myself wrong. It can't be sex-linked dominant. All right, so it's an autosomal dominant situation. Autosomal dominant, I believe, would be best for this one. So, hey, and if you spend a little more time with it, maybe you can discuss it with me. But there you go. So sometimes you have to just work your way through a pedigree to see what works and what doesn't work. Once you find something that doesn't work, then you know it can't be that mode of inheritance. All right, guys. So thanks for listening, and work your problems.